You should not be afraid to start something really big. I think we're all being touched by cancer. Any type of cancer can be potentially treated with this type of technology. The postdoc fellow who initiated the project, William Nyberg, from the moment we saw the data, I told him this is going to be game-changing. In vivo site-specific engineering is possible. CAR T-cell is engineered immune cells. We are adding a new DNA code in the cell, and the goal is to provide an ability to recognize the tumor cells, kill it, and orchestrate the immune system to help combat the tumor. One cell can become a whole army of cells. That's why we're calling it a living drug, because it has this ability to proliferate and persist potentially for years. The current way we're manufacturing CAR T cells is complex. You need to harvest the patient T cells. You draw their blood, you take that immune cells, and then you go to a manufacturing facility, and those are expensive to build. This whole process takes multiple weeks, and currently most CAR T-cell products cost between $400,000 and $600,000 a dose. One way that we're thinking of solving these issues is not taking the cells out of the body and engineer them directly within the body. And injecting a vector that can find T cells and deliver the CAR gene, making the CAR T cells within the body without having this whole manufacturing. It would be what we call an off-the-shelf therapy. Pierre-Louis Bernard, postdoctoral fellow, really helped accelerating the whole project. He started a massive operation here to produce enormous amount of those vectors. This work where we've been able for the first time to place this large piece of DNA encoding a CAR into one very specific place into a T cell in vivo without having to take the cells out. From the first minute we knew that this is going to be big. And it was not only working a little bit, it worked incredibly well the first time around. When we do a single injection of the two vectors here, you see that the tumor initially grow and then completely disappear. And so many other therapies could potentially use the same type of technologies. Here, we're working with the immune system. So there is no chemotherapy that is needed for this type of approach. I definitely lost people that I love to cancer and other disease. And especially now seeing that some medicine that could have saved them are being developed. It's always hard because, like, what if they'll be still around us for a few more years? We are laser focused on going as fast as we can to the clinic to provide those innovations to patients. The drugs in the labs are working so much better now. I really believe there's a bright future that in the next 10 years, there will be new waves of medicines that can be curative in a really large range of disease.